Hi everyone, welcome to researchmd.com. Another great presentation. And let me introduce myself. My name is Dr. PRAMIL. I'm a physician practicing in the United States, program director, internal medicine residency, transitional residency, and I teach medical students and residents on a daily basis. Also, director of research and associate professor of medicine, two large medical schools. Now, we started our lectures on antibiotics. So today we're going to focus about the bacteria and how do they get their energy, right? Anytime a bacteria need to attack a human, right, human body or enter into the cell, they need a lot of energy to attack the organs or replicate itself. Okay, the main thing when we talk about today's topic, how does it produce the protein? That is the main topic. Before we go, when we talk about the bacteria and their energies, so let's talk about what are the raw products needed and all that. Okay, so if you look at this, the building blocks. What are the building blocks needed? You got amino acid and the RNA. When you talk about the energy, there is ATP and a proton motive force. And energy is kind of stored like a protein motive force. Okay, what is proton motive force? The force that promotes movement of proteins across the membrane, downhill electrochemical potential. That's another source of energy, we could say that. Okay, now then depends upon the oxygen use. There's anaerobes and aerobes are there. Okay, the next thing, let's do the classification based on the oxygen requirement. Okay, the first one is aerobes. What do they need? They need oxygen to survive, and then you got anaerobe. They do not require oxygen. Okay, remember that those two. So aerobes, you need to require oxygen. Anaerobes, you don't require oxygen. Now, let's look at some examples of aerobes. So you got Mycobacterium tuberculosis, right? And the nocardia, you got lactobacillus and all that. Now, what are some examples of anaerobes? You got Bacteroid fragilis, Fusobacterium. Those are the two main examples. Now, let's go deep in there, and there's some more classification. Let's look at anaerobes. What are the classification, right? We talk about, I'm going to talk about aerobes first and look at the classification. When you look at the aerobes, we know they need oxygen. Okay, the next classification, there's something called obligate aerobes. Okay, what does that mean? You definitely need oxygen, otherwise they're going to die. What is an example? Mycobacterium tuberculosis, my friend. Okay. Now, the next one is microaerophilic. That means you need only a little bit of oxygen. Okay, main example would be H. pylori. Everybody got that? Now, let's look at the anaerobic again, the big definition. They do not require oxygen. And there's something called obligate anaerobe. They cannot survive with oxygen. Because they cannot survive with oxygen, they will die. Okay, so they are like Clostridium is a good example for that. Okay, then you got facultative anaerobes. So let's look at the definition. They can grow without oxygen, but can use oxygen if present. So an example would be E. coli, the common you see in the urinary tract infection and all that, right? So everybody got that, the classification, we talk about the building blocks, and we talk about the energy, we talk about the oxygen, we talk about the difference between aerobes and anaerobes. Now let's look into how do we produce the protein. It's very important, okay? So main thing, you have to convert this DNA of the bacteria is converted into mRNA. That process is called what? Transcription. Okay. What is the enzyme? Very, very important. You got RNA polymerase, and they use the ribonucleic acid and convert it into mRNA. Okay. That process is called what? Transcription. Okay. Now there's another big process called this mRNA is converted into actual protein. That's what we call translation. Okay, when you use the translation, there's two products here. You need a ribosome and you need tRNA and the amino acid. All of those kind of play a very big part making this mRNA into the protein. Okay, very important, the ribosome. So let's look at the structure of the ribosome, a little bit different. Bacterial ribosome is different than the human ribosome. When you talk about bacterial ribosome, you got two subunits. One is 50S subunits and a 30S subunit, and they make together a 70S subunit. Okay? When you talk about the 50S subunit, you got two rRNA molecules and 34 proteins in the 50S subunit. And in the 30S subunits, you got one rRNA molecules and 21 proteins are there. So that is the structure of the ribosome. When you talk about human, it's like 80S, which is a combination of 60S plus like 40S. Okay, that's a little bit different. Why is it important? So a lot of antibiotic we use, kind of based on like against attacking the ribosome so they cannot survive. Okay. So remember to make sure, like we know, I'm just going to summarize a little bit. Look at the raw products, amino acid, tRNA, remember about APP, ATP, and proton mode T-force, and then oxygen requirement, aerobes and anaerobes, and look at the classification. Aerobes require oxygen, anaerobes does not require oxygen. And then we come up here, first step is the transcription, the DNA is converted into mRNA, and then translation, mRNA is converted into protein with the help of ribosome and tRNA, and then we have a nice structure of the ribosome, which is 50S, 30S, and a combination to 70S ribosome, okay? So we can make antibiotics to attack this so they can prevent or destroy them, okay? Thank you so much for watching. We'll be back with another presentation soon. Thank you.